way back in 2002, I remember. I'm forgetting his name. So the art trustee or the founder of this university, Sayyid Babar Ali, he was on a visit to MIT. And the president of MIT told him that if you want to go to sciences, you have to bring in interdisciplinary school of science
Yeah.
and engineering.
Oh,
Uh, and the philosophy of school is no boundaries philosophy. So engineers, biologists, chemists, everybody comes in and they have to have first year core courses. And we have a very strong advisory board. You may know Professor Bob Jaffe from MIT. He's a professor of physics in
yes, I
MIT.
do. Yeah, we
Yeah,
know
so
about.
he's on our advisory board, and there are many other professors. Rick Larson from MIT as well. He was in operational research at MIT. So
Yeah.
they were the founding advisory board of our
Oh,
school.
excellent.
Yeah.
Well, I've only been at MIT on and off since 1960, so it's been a couple of years. Just a wink in the eye of the Lord, but for me, a major chunk of my lifetime.
Yeah. So looking at your profile, I was talking to Faisal that, Professor Weinberg is there before we were even born. So you did your PhD.
Well, that makes me feel great.
So such a rich experience in the field you have.
Well, by coincidence, I could witness from already early in my career the whole development of the modern field of molecular cancer research. Getting old has only one perquisite, one advantage these days, and that is one gets one's COVID shots early.
Mhm.
Because old people, as you must know, are very susceptible to the disease. So I'm now immune for the first time and only gradually emerging from COVID exile.
Mhm. So you had both the shots or
Both shots of Pfizer, yes.
yeah, Pfizer?
Exactly a year ago, my wife and I, we retreated to our cabin deep in the woods of New Hampshire, north of Massachusetts, for 9 months just to get away from humanity. And then only recently came back to the Boston area, where my research institute, where MIT is located.
So did you experience some headache or some after effects of the vaccine?
I did not. My wife was laid up in bed for a day after the vaccine. So I guess it means she has a very active immune system. We don't really know how everything works. It will be interesting to know how we deal with this virus in the future. I suspect in the longer term, it will become a virus, an endemic virus, much like flu. So that we have yearly epidemics of COVID, and we might need to get booster shots every year against new versions of the COVID virus. Another complication in our already complicated lives. Mr. Jahar, when do you usually begin these talks? Right on the hour, or you give a couple minutes of grace time? What is your usual practice?
So we usually start about so considering this talk, we can start it at 9 a.m. exact of your Eastern time, and
Okay.
and we normally start it about like for like 5 minutes over, depending upon how much audience we have. I can see a number of people today, so I think we can start on time today. And
It's up to you. And I find long introductions, Mr. Jauhar, very embarrassing. So if you just made a very short and truncated introduction, my favorite introduction is, here's Professor Weinberg. He's a great human being in his own mind, but that doesn't work well in certain audiences. So we'll do whatever you want. Okay, but a very short introduction would be gratefully received by me. Thank you.
okay, sure, definitely.
And I imagine your own travel has been also strongly curtailed by the COVID pandemic.
Yes. Yes. So the most unfortunate thing was that we had to shut down research for nearly 6 months. I think that was,
Well, it was not much different in Cambridge, Massachusetts, I will tell you that.
uh,
People have now gotten into the lab, but with a certain degree of distancing. One is supposed to keep 2 meters apart from one another.
and how was that last year?
Well, there were people allowed in the lab, but only one person per room, or sometimes 2 persons per room. And they had this rule, you should never get closer than 6 feet to another person. Do you know where the number 6 feet came from? Somebody picked it out of the air. No scientific basis whatsoever. Everybody followed it slavishly, but no scientific basis for 6 feet. But anyhow, here we are. Okay.
I think we can start.
I don't see many students, but I guess some of them will still come in later.
Yes, so they will be joining us soon. We have nearly around, I think, 33 students right now, and some will join via Facebook live stream as well.
Okay.
So have the days started to get a bit warmer at your end?
Yes, yes, we still have snow here and there, and there's still frost on in the ground. You don't know what that's like, but here the ground can freeze to 4 feet below the surface, and it takes a whole month to thaw out,
Oh,
which generates what's called mud season. Mud season is when the ground underneath is still frozen and the ground at the surface is thawed, and the water at the surface can no longer escape because of the ice below it. So it's one of those things. Of course, we don't have monsoons, so you're blessed with that. Actually, are there monsoons are more typical of Bangladesh than they are of Pakistan, or both?
Yeah.
Both are equally affected.
yes, so not as compared to Bangladesh, it's very severe at that side. We
Yeah.
get a lot of moderate here beginning June and July.
Got it.
But we also get 40 degrees
Ooh.
Celsius over here. So
Okay, well, I'm ready to go if you'd like me to go.
okay, so I think we can start. Sir can introduce. Dr. Tariq, you can introduce and we can start then.
Okay. So it's a real pleasure and an honor. And I would, in the words of Professor Weinberg, I think we will have short intro. I think the best intro is here we have Professor Weinberg with us.
Perfect.
A great human indeed. Thank you for joining us, Professor Weinberg. Over to you.
Thank you for having me. Is my PowerPoint coming on the screen?
Yes.
Yeah.
Title page is on the screen, okay?
Yeah.
Yes.
Yes? Okay, I will therefore begin, and we'll have some questions and answers afterwards if there were interest. So I'm dealing today with the molecular mechanisms of cancer formation. Many of you may know that cancer formation is a multi-step process, here seen in at the histopathological level in the context of colorectal carcinoma, a whole series of distinct stages. And we know that these multiple distinct stages operate during the pathogenesis of a whole series of distinct kinds of human tumors, these being a collection of half a dozen distinct types of carcinomas. But this multi-step process also operates in a variety of other tissues as well. The process as a whole, you may wish to know, takes a long time. So here's the only accurate measure we have of how long it takes to actually induce cancer. This being given by global cigarette consumption in the 20th century and the uptick in lung cancer deaths. And this had an exactly 30 years lag time. So from this, one can begin to imagine that the formation of many kinds of human tumors actually occupies a multiple series of decades, 3 decades, 4 decades, 5 decades. And if we're lucky, there may be incipient tumors growing in our tissues that never actually become clinically apparent during our lifetimes. Already in 1989, the laboratory of Bert Vogelstein at Johns Hopkins University in Maryland began to describe the fact that as one had these histopathological progressions seen here below, one could see a series of changes in the loss of heterozygosity in various chromosomal sites. You see that they're indicated here. At one of these stages, one found the activation of a KRAS oncogene. And this began to indicate that there was actually the mutation of multiple genes. Here we see these genes now named, and that as cancer cells progressed from full normalcy to at least primary tumor formation, there were a series of somatic mutations that propelled these cells from one stage to the next to form increasingly aggressive tumors. Moreover, there are other changes in the structure of the DNA, chemical structures, such as the methylation of CpG dinucleotides. The scheme that was evolved was a series of steps where each mutation that a cell sustained led to a clonal expansion of its descendants, one of which in turn could then acquire yet another mutation. And this began to suggest a series of successive clonal expansions that in aggregate ended up leading to a primary tumor. And in fact, we know how many such mutations are required. Here's some work that was done in my lab in 1999, which suggested that one needed to introduce at least 5 different changes into a human cell experimentally in order to transform it into a tumor cell. These 5 representing 5 different regulatory pathways within a cell. And by the way, in mice, you only need 2 changes. So our cells are wired up to be different than those of rodents, very different. And these changes in aggregate lead the conversion of a normal epithelial cell in the context, let's say, of the colorectal cancer into a transformed cell, in this case, presumed to be a primary tumor. But this raises an interesting question, an important question, and that is, are these somatic mutations, which I've indicated here in the red arrows, are they the only change that cancer cells need to undergo en route to becoming, for example, invasive and metastatic? How important are non-genetic programs in determining the biology of cancer cells, one might ask? And so this is something I wish to focus on today, that is to say, the non-genetic programs. We know that they're important, these epigenetic programs, because we know that stem cells can differentiate into a whole variety of different cell lineages without undergoing a change in the nucleotide sequence of the DNA, and therefore all of these differentiation programs are by definition epigenetic. One important question, by the way, is the continued influence of the differentiation program of the normal cell of origin. In other words, the original normal cell that participated as the ancestor of all of the subsequently arising cancer cells. And here in this transgenic model of a mouse lung cancer, one created 2 different kinds of primary tumors. And these primary tumors were in adjacent cells within the context of the lung. And if one compared their transcriptomes, that is the array of genes that were upregulated or downregulated in these tumors, one saw that in fact they were quite different in terms of the relative transcriptional programs. And this began to suggest that the normal cells of origin continued to influence the behavior of the derived tumors. In other words, that the differentiation programs of the normal cells of origin represented an important factor in creating the particular kinds of phenotypes that one associates with the cancer cell. And given that, one comes to realize here that this normal epithelial cell, whether it has one kind of differentiation program or another, may continue to exert its influence on later stages. It's not as if the differentiation program, the pre-existing differentiation program operating here as an epigenetic program, continues to impact the biology of the derived cancer cells many steps later, maybe even many decades later. So this is one example of where a non-genetic influence is very important in determining some of the aspects of a cancer cell. And we really understand very little about the differentiation program of the normal cell of origin, that is the cell that would be located down here to the left. There is, however, yet another question, and that is, granting everything that I've just shown you, what happens here? Here we have a primary cancer cell, we have invasion and metastasis. And as I will argue, when you go from primary cancer cell to invasion and metastasis, this probably does not involve additional somatic mutations, but might instead represent another epigenetic program, which must cause us to recalibrate our thinking. In fact, this last step here from carcinoma to invasion, it's sometimes called the invasion metastasis cascade. And what one sees here is that this last step actually subsumes a whole series of substeps that are laid out here graphically and lead from the initial primary tumor to intravasation into blood vessels, extravasation, escape into the parenchyma of distant tissues, and ultimately the outgrowth of micrometastatic colonies into macroscopic metastases. And the question is, given all of these traits, how do cancer cells acquire the multiple distinct capabilities that are required in aggregate in order for a cancer cell to successfully found a metastatic colony at distant sites in the tissue? And one answer to this question is given in this micrograph here, where we begin to see that we have here implanted carcinoma cells. They're human carcinoma cells in a immunocompromised host mouse. Here we see the carcinoma cells expressing the hallmark epithelial marker cytokeratin in red. Down here, we see other carcinoma cells at the outer edge expressing the human vimentin, which is a mesenchymal marker. And on the outside, by the way, are the stromal cells recruited from the mouse host. And thus we begin to see here that these cells in the carcinoma, and these are, the green cells are, by the way, also from derived initially from the red cells, have undergone some profound shift in their cell biological program. And this begins to suggest, it doesn't prove yet, that here we have a transdifferentiation program from initial epithelial cells, indicated here in red, to subsequently arising mesenchymal cells, indicated here in green. It's not yet proven by what I've shown you, but one gets more suggestions about the correctness of this notion when one looks at these tongues of carcinoma cells. Here, once again, one sees such an implanted carcinoma cell. These cells here are expressing cytokeratins, once again, the hallmark, the keystone of the epithelial cell state. On the outside, once again, are mouse stromal cells recruited to the tumor. But here we see a rim of cells on the outside that has shut down the cytokeratin expression. Here's yet another tongue of carcinoma cells. And we see here in this micrograph that the cells on the outside of this tongue of carcinoma cells now have begun to express vimentin, that is a mesenchymal marker. And therefore these cells have moved from an epithelial to a mesenchymal state. And that such a shift represents an epigenetic program called the epithelial mesenchymal transition. Ostensibly, the epithelial mesenchymal transition is influenced by the neighborhood of the cells, because here we see that the cells that have acquired a mesenchymal phenotype are adjacent to the recruited mouse stromal cells. And this suggests that there are contextual signals that have influenced the phenotype of these cells. And here one can see at a mechanistic level what happens at this interface between the epithelial carcinoma cells and the stromal cells, these cells having undergone an EMT. Here's one example as done by Ann Lee, where here we have a carcinoma cell in light purple. This carcinoma cell is releasing interleukin-1, which many carcinoma cells do. And this interleukin-1 impinges on cells within the recruited stroma, mesenchymal stem cells or fibroblasts in the stroma. And the mesenchymal stem cells respond to the interleukin-1 through this heterotypic signaling by releasing reciprocally a whole series of other signals back on the carcinoma cells, including these interleukins here, GRO alpha, and even prostaglandin E2. And the signals impinging back on the carcinoma cell ultimately induce the carcinoma cell to activate its previously latent EMT program, indicating that the carcinoma cell is activating a previously silent epigenetic program. And this program was first described by Elizabeth Hay more than 30 years ago as the epithelial mesenchymal transition. I just mentioned it, and it is a multifaceted program in which cells undergo a profound shift in phenotype, moving from an epithelial to a mesenchymal state and acquiring a whole series of distinctive markers, as you can see here. In truth, there are probably 400 genes that become upregulated or downregulated in response to this program. So it is a coordinated, multifaceted program, which is hardwired, it seems, in all epithelial cells throughout the body. And again, I would emphasize that cells like this light purple one here don't spontaneously activate the EMT. Instead, they do so only in response to signals that they've received from other cells in the tissue. In this case, let's imagine stromal cells that may be nearby in a recruited tumor. Now, in fact, I would also mention the fact that there's not a single EMT program. There's a whole variety of EMT programs, different versions of a core EMT program, and these different versions may be manifest in different tissues, depending on pre-existing differentiation programs, and depending on, for example, the somatic mutations that cells may have acquired during the course of multi-step tumorigenesis. Moreover, I would mention the fact that the EMT program is of ancient vintage. We know that all metazoa express a set of transcription factors, they're abbreviated EMTTFs, a set of transcription factors at various stages during their embryogenesis. You can see them here. And in fact, this array of transcription factors, which are indicated here, snail, slug, twist, goosecoid, etc., is present in the genomes of all metazoa, indicating that these transcription factors and the genes encoding them were already developed 500 or 600 million years ago, prior to the evolutionary radiation that led to the development of a whole variety of different kinds of metazoan phyla. And these have been highly conserved, such that a transcription factor, an EMTTF from one phylum, can actually operate in the cells of another phylum. Clearly, they've been retained, these transcription factors, because they're critical to normal embryogenesis, and moreover are important for the morphogenetic steps that lead to successful completion of a complex developmental program. Here we see, for example, that the twist EMTTF is expressed in a set of cells in a recently fertilized Drosophila egg. And here we see in this embryo the fact that there's introgression of these cells, the brown cells, forming this kind of the initiation of gastrulation. And thus we see the twist induced by inducing an EMT is, in this example, for example, very important in gastrulation, which we all recognize is a critical component of the formation of the multi-layered tissues that are present in all metazoa. I want now for the moment to shift gears to what seems to be an unrelated topic, and that is the question of cancer stem cells. Here we see the work of Mohammed Al-Hajj and Michael Clark, 2004, in which they reported that they could fractionate cells from a human breast cancer cell population, each dot here representing a single cell, into two distinct subpopulations, doing so on the basis of cell surface expression of these two cell surface antigens, CD44 and 24. And when they did that, they found that cells over here, as few as 200 of these could form a new tumor. Whereas, in contradistinction, the cells in the majority population, as many as 20,000 of these, when they were implanted, they even failed to form a new tumor, indicative of the fact that these two populations resided in distinct states, distinct epigenetic states, because the two populations of cells were genetically identical. And so here, once again, we have a shift or a residence in two alternative epigenetic states, phenotypic states, which in this case seems to empower certain cells to seed new tumors upon experimental implantation, and conversely prevent other cells from doing so. Here's yet another representation of this fractionation of breast cancer cells using the same cell surface markers, albeit in a slightly different convention, as performed by people in my own laboratory. Here are the 44 high, 24 low cells, and here conversely are the 44 low, 24 high cells. Here's where the stem cells would reside. Here's the majority population of non-stem cells. To the extent that there are tumor initiating cells in this population, they would be up here in the upper left. To the extent that there are non-tumor initiating cells, they would almost all of them would be here in the lower right. And this begins to invoke, obviously, the notion of a hierarchical organization that is often used to depict the organization of normal epithelial tissues, in which one has self-renewing stem cells here, transit amplifying or progenitor cells, and ultimately post-mitotic differentiated cells. These would be the stem cells, and these would be the non-stem cells in such a normal tissue. And this kind of scheme has been appropriated by cancer researchers to help explain the phenomenon I've just described to you here as well. Again, these cells up here at the apex of this hierarchy are capable of seeding a new tumor when implanted experimentally. The bulk of the cells have failed to induce a new tumor, indicative of the fact that as they differentiate in this direction, among other changes, they give up the opportunity of being tumor initiating cells. Some years ago, two people in my own lab, Senderoy Mani and Wenjun Guo, posed the following question. And why they posed this question, I do not know to this day. Some idea occurred to them, and that was to pose the following question. Is there any connection between the EMT program that I described before and the stem cell program that I just mentioned now? And so they took two alternative EMT-inducing transcription factors, the aforementioned snail or twist, and they introduced one or the other into cells of this population and asked what the consequences would be. And what happened was that such cells, which normally look epithelial, if one looks at the bulk population, if snail or twist were introduced into them, these cells now assumed a new morphology, a morphology reminiscent of more mesenchymal cells. At the same time, what they observed was that the population of cells that lay here in the lower right, they started migrating en masse to the stem cell position. And this provided the first suggestion that there was an intimate interconnection between the EMT program on the one hand and the stem cell program on the other. Here we see that this interconnection between the EMT program and the stem cell program also extends to normal epithelial cells. Here in the work of Wenjun Guo is a cross-section of a normal mouse mammary gland. The previous work I showed was about cancer stem cells. Now I'm talking about normal stem cells. What we see is that the normal mouse mammary gland represents a bilayered epithelium, a layer of luminal cells confronting the milk-producing duct, the cavity in the middle of the normal mouse mammary duct. And on the outside is a basal layer of cells, and the stem cells within the basal layer are known to, or the stem cells are known to reside in the basal layer of these ducts. And what was shown here by Wenjun Guo is that the slug EMT-inducing transcription factor was expressed continuously tonically in the basal cells of these mammary ducts, testifying to the fact that the EMT program, at least as manifested by this particular EMT-inducing transcription factor, is operating tonically, continuously in these normal cells here, which may in fact be stem cells. In fact, Wenjun Guo could actually demonstrate an association experimentally between the activation of the EMT program and the acquisition of normal mammary stemness. Thus, he could do as had been done for half a century before. He could remove the incipient epithelial cells from the mammary stromal fat pad of a young female mouse. And as in doing so, one therefore eliminated all of the cells that could seed a mammary duct. But in this cleared mammary stromal fat pad, one could then introduce experimentally candidate mammary stem cells. And if the candidate mammary stem cells were indeed functioning as stem cells, they could generate an entire mammary ductal tree, which resembles at many levels a normally generated mammary ductal tree seen here on the left. This obviously represents a stringent test of stemness, because here, if there are mammary epithelial stem cells that are actually present in the implanted cells, then they're capable of generating an entire mammary ductal tree, i.e. an entire normal adult organ, because these reconstituted mammary ductal trees seeded by implanted mammary cells are functionally, morphologically identical to those that are generated by normal developmental programs in a young adolescent mouse. And here's an experiment that demonstrated experimentally that in normal mammary epithelial cells, there was a tight interconnection between stemness and the EMT program. Here is the result of implanting 10,000 normal mammary epithelial cells in a cleared mammary stromal fat pad. Nothing happened. Here is the result of taking an equal number of cells, inducing them transiently to enter into a more mesenchymal state through the transient expression of the slug EMT-inducing transcription factor, which was thereafter, by the way, shut down, implanting those cells that had transiently experienced an EMT program in the mammary stromal fat pad. And 6 weeks later, one sees that as few as 100 of these cells that historically had experienced the EMT program under the influence of the slug EMT-inducing transcription factor, now as few as 100 of these cells could seed an entire mammary ductal tree, indicating that there was more than 100-fold increase in the formation of normal mammary stem cells as a consequence of transient exposure of the ancestors of these cells to the EMT-inducing transcription factor slug together with SOX9. And we know from later work, by the way, that the expression of slug is found in a whole series of normal stratified epithelial tissues throughout the body. Ostensibly, these are normal stem cells in these bilayered epithelial, but to date, we've not had any indication that there are actually, that slug is expressed in single layered epithelial, so we don't understand what governs the behavior of the stem cells in these single layered epithelia. Nonetheless, this association between EMT and stemness also is related to cancer stem cells, which are defined as the ability to seed new tumors upon implantation in appropriate mouse hosts. Here we see cells, the work of Chenier, cells which express high levels of the snail EMT-inducing transcription factor. This is now in cancer cells, not normal cells. And these cells here, 1 in 91 of them can seed an entire new tumor. But cells that lack snail in the second row here, but express slug, 1 in 72,000 of these cells can seed a new tumor. And so there's an 800-fold difference in the representation of tumor initiating cells in these two subtypes of cells within, let's say, a mammary, a tumor-prone mouse, a transgenic mouse. And so we begin to see this association between the expression of an EMTTF, in this case, snail, not slug, but snail, and the acquisition of tumor initiating potential as judged by implantation at various limiting dilutions in an appropriate mouse host. And here's another manifestation of this. These are human breast cancer cells. They're called MCF7 RAS cells, indicated up here. And these cells are generally non-metastatic. And so if one implants them and looks at the primary tumor 6 or 8 weeks later, then one sees that the primary tumors that result have a rather smooth outer edge, indicative of the fact that these cells are non-invasive. On the other hand, one can take the same population of cells, induce transiently the expression of these two EMT-inducing transcription factors, shut down these EMT-inducing transcription factors, and then wait another 6 or 8 weeks. And now one sees that the outer edges of the resulting primary tumors behave very aggressively, because now obviously the carcinoma cells are invading into an adjacent mammary fat pad. And so these cells, 8 or 10 weeks after the initial induction of slug and SOX9, remember that their ancestors had experienced an EMT program and as a consequence have become quite malignant. Perhaps even more interesting are the ability of these carcinoma cells, the MCF7 RAS cells, to form tumors in the lung following, let's say, subcutaneous implantation. Here are control MCF7 cells, which form neither macro nor micro metastases in the lungs, obviously by metastatic dissemination. But for example, if these cells had been exposed transiently to this combination of EMT-inducing transcription factors, here slug and SOX9, then on average, these mice would form 100 macroscopic metastases and 400 micrometastases in the lungs of mice. And this begins to demonstrate quite dramatically that there's a causal relationship between the transient expression of an EMT-inducing transcription factor and the ability of cells to actually acquire metastatic and aggressive malignant phenotypes. And experiments like this begin to suggest the following simple mechanistic model: that when cells progress, as I showed you before, up to the stage of primary carcinoma, if one turns on in these primary carcinoma cells the EMT program, that suffices without additional mutations to enable cells to physically disseminate to distant tissues throughout the body and form micrometastases. That is to say, to enable these cells to complete most of the steps of the invasion metastasis cascade. However, the last step, to my mind, represents a bit of a mystery, because when these carcinoma cells arrive in a parenchyma or distant tissue, they're ill-suited at the moment of arrival to form a robustly growing tumor. And as a consequence, they must undergo some kind of adaptation to enable them to thrive in a distant tissue. After all, why should a mammary cell be able to grow in the context of the bone marrow or the brain or the lungs? And so to my mind, this remains a major unsolved problem. How do cancer cells adapt to a distant tissue? To my mind also, the ability of these cells to physically disseminate can be nicely explained by the actions of the EMT program. The proofs of this, not all of which I'm showing to you here today. So this begins to indicate that by focusing exclusively on somatic mutations, I mentioned them before, one misses out on understanding a critical component of the phenotypic shifts that occur as cancer cells progress to high-grade malignancy. And this also calls into question the monomaniacal study of cancer genomes, which have been pursued over the last 2 decades, because as you might intuit from what I've just shown you, genetic changes in the genomes of cancer cells do not address the complexity, do not address the problem of metastatic dissemination. And again, here is the problem, as I've mentioned before, the last step of colonization, which represents the ability of cells from one tissue of origin to metastasize and form sites of metastatic colony formation in distant tissues. Here you see, depending on the width of the arrows, that, as you know, different primary carcinoma cells have abilities to differing extents to form tumors in distant sites in the body. And in many cases, the anatomical layout of the circulatory system, such as is the case here with prostate cancer, doesn't explain the proclivity, the propensity of primary carcinoma cells to form tumors in distant tissues, that is, to colonize those tissues. Again, to my mind, this remains a challenging question. I would just mention, by the way, that in fact the EMT program does not act as a binary switch, as you might have concluded from what I've said until now. That is to say, if here we have epithelial carcinoma cells, and here are highly mesenchymal carcinoma cells, it's not as if the EMT program causes cells to move from one state all the way over to the other, from E over to M. Instead, it's almost always the case, and possibly always the case, that to the extent that carcinoma cells activate a previously latent EMT program, they advance to differing extents in the direction of being mesenchymal, but never become entirely mesenchymal, instead residing in a whole series of intermediate states, which one can in aggregate say are quasi-mesenchymal. And these represent the products of the EMT program. And in truth, we don't really understand how many distinct phenotypic states there are, intermediate states, quantal states that separate the epithelial and the mesenchymal state that one might imagine would occur if it were the case, which it is not, that the EMT program operates as a binary switch. And I just show you a visual emphasis of this. Here's a section of a human breast cancer cell population. And what we see here, without going into any details, is that many of these states reside in to different degrees in quasi-mesenchymal intermediate states that lie halfway between epithelial and mesenchymal, halfway between E and M. And this has prompted, among others, Brian Beery to begin to try to depict or catalog what happens when a cell activates the EMT program and moves from E toward M. And what one sees is that as cells become increasingly mesenchymal, and as the EMT program is increasingly played out, cells lose E-cadherin, which knits together laterally the epithelial cells in an epithelial cell sheet. Then the CD44 cell surface marker is turned on. Then vimentin is turned on. The P63 transcription factor is eventually shut off, as is in this case integrin beta 4. And I would just mention the fact that I cited before, spontaneously arising human cancer cells never make their way all the way over to the mesenchymal state. Instead, cells venture to differing extents into an intermediate set of phenotypic states indicated here. And if one looks at where tumor initiating cell frequency can be found, TIC is the same as cancer stem cell frequency. What one found, as Brian Beery indicated here, is that the stem cells seen here in these purple dashed lines actually reside in an intermediate state, a quasi-mesenchymal state that is neither fully epithelial nor fully mesenchymal. And there have been, and here we see that 1 in 3,000 cells can initiate a new tumor in this particular experimental model, whereas cells that have ventured all the way over here toward the mesenchymal state, now only 1 in 750,000 cells can seed a new tumor. So here we see a 250-fold difference in relative tumor initiating ability. And there have been papers that have claimed triumphantly that the EMT program cannot be associated with the stem cell program because in certain experiments, if one induces the EMT program, cancer cells actually lose their stemness, their tumor initiating capability. Here, what I'm showing you is that in such experiments, one had turned on the EMT program excessively and pushed cells all the way over to here, where, as you might imagine from what I've depicted here, cells actually exhibit a loss of stemness, a loss of the ability to seed a new tumor. And by the way, there's a great degree of phenotypic plasticity. That is to say, cells can move from E over to M, and cells in these intermediate states can move back and forth, alternating between distinct phenotypic states that lie along the epithelial to mesenchymal spectrum. In truth, and of course, I'd only be truthful with you, there are other components of the EMT program that make it critically important to clinical cancer. For example, cells that have activated an EMT program move from a chemosensitive often to a chemo-resistant states. The self-renewal capability, that is to say stemness, is also associated with the EMT program, as is, by the way, the ability to seed new tumors. And so this EMT program assumes a major role in explaining malignant progression, at least of carcinoma cells, probably also of brain tumors, which obviously also derive from a primitive epithelial cell sheet in the early organ, in the early embryo, but other kinds of tumors, such as hematopoietic tumors and mesenchymal tumors, such as sarcomas, we have no idea actually what program influences them to become metastatic, and whether the EMT program contributes at all to their malignant acquisition of malignant phenotypes. Should I end here or should I continue? Mr. Jauhar, please give me an information. Here are the good-looking people in my lab, but I need to know how the time is for you. Would you like?
So we can continue for the next 10-15 minutes.
Okay, so I'll continue. You'll see this picture again, and once again, you can admire how good-looking the people are in my lab. Anyhow, so here's an interesting question. If we deal with the whole issue of immunotherapy, that is to say, if we try to eliminate tumors, how does the phenotypic state of the carcinoma cells affect their responsiveness to elimination by checkpoint immunotherapy? And checkpoint immunotherapy represents a very successful therapy for certain kinds of tumors. It represents using monoclonal antibodies to block several negative feedback mechanisms that prevent an overly aggressive attack on carcinoma cells by cytotoxic lymphocytes indicated here below. And here one can actually block the one of these negative feedback mechanisms through the use of an anti-CTLA-4 mechanism and anti-CTLA-4 monoclonal antibody. We could get into the details here, but we needn't do so. And let's just mention that the successes of this checkpoint immunotherapy have been stimulated largely in the context of metastatic melanoma, where these two monoclonal antibodies that block PD-1 and CTLA-4, they actually have created cures in a number of people, many people, using this checkpoint immunotherapy in individuals who were otherwise destined to die quickly because of metastatic melanoma, as is seen here in this purple curve. So survival curves like this are highly suggestive that these individuals, many months later, have actually been cured of their tumors through this checkpoint immunotherapy. And yet it's sadly the case that checkpoint immunotherapies have not proven to be that successful in the context of a whole series of carcinomas, raising the question of who actually is responsible, or what agents are responsible for the failures of checkpoint immunotherapy in treating many carcinomas. Here, I just show you the track record. Metastatic melanoma responds to certain kinds of checkpoint immunotherapies and not to other kinds of checkpoint immunotherapies. And to pursue this, Anushka Dongre from faraway Mumbai, but she's now in my lab, she undertook a series of studies with two sets of mammary carcinoma cell lines from transgenic mice, from the transgenic MMTV polyoma middle T model. And these are two cell lines that were derived from one set of tumors or another set of tumors. And here, these two clonal populations, these cells called epi here, remain in an epithelial phenotypic state. These cells over here, called mesenchymal, remain in a mesenchymal state. And for reasons we don't understand, these cells remain stably ensconced, stably established in one phenotypic state or the other. We don't understand why there's not some kind of phenotypic plasticity, but for whatever reason there isn't, and this represents a great experimental convenience. And so Anushka Dongre undertook to determine whether the immune environment of the epithelial and the mesenchymal cells represented an important determinant of whether or not these cells were susceptible to elimination by checkpoint immunotherapy. And what she found was, when she began to look for a whole series of stromal cells and immunocytes, is that immunosuppressive macrophages, here with one pair of these tumors, or another pair of these tumors, the immune suppressive macrophages were present in much higher levels in the stroma of the mesenchymal tumors, whereas the epithelial tumors. And note when I say mesenchymal here, I mean the quasi-mesenchymal cells that reside halfway down the spectrum from E to M. Moreover, if one looked for the markers of immunosuppressive macrophages, then one found that in mesenchymal tumors, there were far more immunosuppressive macrophages, moving here from left to right, than there were in the epithelial tumors. So the whole population of the immunocytes in the cancer-associated stroma was very different. Moreover, cytotoxic T cells, as you know, are important agents of the immune system, which dispatches these cells to kill cancer cells. And the cytotoxic T cells that were present in the mesenchymal tumors were present in much lower numbers than they were in the epithelial tumors, once again indicating some kind of dysfunction in the mesenchymal tumors of the immune state. Moreover, the few T cells that actually ventured into the mesenchymal tumors had far higher amounts of T cell exhaustion as compared with the cells in the epithelial tumors. To put it another way, you may know that CD8 cytotoxic lymphocytes can become functionally exhausted, in which case they no longer are able to execute their cytotoxic powers. And here one sees that the levels of exhausted T cells are far higher in the mesenchymal tumors than they were in the epithelial tumors, once again indicating a measure of dysfunction. There's yet another immunosuppressive cell, you may know, regulatory T cells, and these immunosuppressive lymphocytes are present at far higher levels in the mesenchymal tumors relative to the epithelial tumors. And one can see also that if one looks at these immunosuppressive macrophages, you might be able to see them here in red. They're present in far higher concentrations in the mesenchymal tumors than in the epithelial tumors. In fact, if one looks at the localization of the immunosuppressive macrophages in the epithelial tumors, the M2 immunosuppressive macrophages are exiled to the outer borders of the tumor mass, whereas in the mesenchymal tumors, the immunosuppressive M2 macrophages are welcomed into the tumor mass, as you can see here from these red dots. Moreover, yet another expression of the differences, the profound differences in the immune microenvironment of these tumors is indicated by the MHC class 1 molecules, which, as you know, represent the hands of the immune system that display to the outside world oligopeptides that might in principle represent targets for recognition and attack by CTLA8 cytotoxic lymphocytes. And here we see that these MHC class 1 molecules are present often at far lower levels in the mesenchymal cells than they are in the corresponding epithelial cells. And that occurs through a whole series of different comparisons where one sees that essentially these cells that become mesenchymal have far lower levels of MHC class 1 than do their epithelial counterparts. And not having MHC class 1, in principle, these cells can fly under the immunological radar simply because these cells no longer display to the immune system potentially immunogenic epitopes. And here I indicate a quite dramatic difference in the responsiveness of these various kinds of tumors to this particular brand of anti-CTLA4 checkpoint immunotherapy. Here we see the outgrowth of an epithelial tumor, and if anti-CTLA4 checkpoint immunotherapy is applied, this tumor growth is held down nicely. Here we see, in contrast, cells made by the genetically identical mesenchymal carcinoma cells, and these cells continue to grow out, you see in the solid line, and if one applies checkpoint immunotherapy, they're totally unaffected by this therapy. And this begins to indicate that the phenotype of the carcinoma cells represents an important determinant of their responsiveness to checkpoint immunotherapy. And here I indicate another manifestation of this. One can ask the following question, as Anushka Dongre did. What happens if one mixes therapy-resistant mesenchymal cells with therapy-sensitive epithelial carcinoma cells? So here are the responses of fully mesenchymal tumors to checkpoint immunotherapy, as I showed you before. they are totally unaffected. Here are the responses of fully epithelial carcinoma cells, and they're strongly held back. But one can also make 9-to-1 admixtures. In mixed tumors, one has 1 part of mesenchymal cells and 9 parts of epithelial cells. And what she saw here was that, in fact, the minority of the mesenchymal cells is able to protect the majority of epithelial cells from immune attack and elimination. And this begins to mirror what happens in actual human tumors, where mesenchymal minorities of cells operate and where they may, in principle, exert immunosuppressive effects on the tumors as a whole. And we suspect that this may, again, represent a mirroring of what happens in mice, and may explain the extent to which many human carcinomas are refractory to checkpoint immunotherapy. So I'll end on this note. Here we see the good-looking people in my lab once again, and I'd be very pleased to take questions at this point from Mr. Jauhar and his colleagues, and obviously from the faculty that are present.
Okay, so we'll take questions. So audience, any question before I start asking? Okay,
Please do not be intimidated.
Zen Umar has a question. Zen, you can ask.
So it's been an honor, Professor, to hear your talk live. I was just wondering,
It's my honor to have you come. Thank you.
I was just wondering that would pediatric cancers be considered a deviation from whatever we're establishing in the mainstream cancer research, or will it just encompass pediatric cancer research as well? Because obviously the timescales of development, how they respond to therapy, their immune system in general is all very, very different from adults.
That's absolutely correct. And for me, pediatric cancer represents a profound enigma, a puzzle. I don't really understand how pediatric cancers arise. I told you before, I gave you in passing some evidence that in the case of adult cancers, one needs to perturb 5 distinct regulatory pathways in the cancer cells in order for them to become tumorigenic. But in pediatric cancers, there are far fewer genetic changes that are required for the acquisition of tumorigenicity. And so to my mind, there's an entirely different set of lessons that governs how pediatric cancers are formed. And I'm totally puzzled by that. And just as you said, the lessons about pediatric cancers must be generated in an entirely independent line of research, which is not well informed by what we know about adult cancers, again, for reasons that remain a mystery to this day.
Thank you.
Okay, Dr. Safi has a question. Dr. Safi, you can go on.
Yeah, so Bob, it's such a pleasure to be hearing from you directly. I have been a long-time fan of your work, and
it's a pleasure for me to be in Lahore. I've never been there before, so now I am.
yeah, I wish we
For
could
having me.
halwa puri and lassi, but we'll keep that for another time. So I have a couple of inquiries. So the first one is, how do you see the multi-scale modeling and patient-centric models contributing to the advancement of cancer research? And secondly, if you may, then how would RNA-seq snapshot databases be best utilized to study, the temporal evolution of cancers?
Well, let's go to the questions one at a time. The fact is, over the last 2 decades, as you well know, there's been the accumulation of an enormous data set of genetic mutations that have acquired in human tumors. There's more than 200 genes that can become mutated, and there are large data banks of the genomes or the genetic aberrations that are present in different subsets of human tumors. The question is, having gotten these large data sets, has that led us directly, as day follows night, into being able to treat these tumors? The presumption, the hope was, at least the hope expressed by the people who actually did this work, or told us that we should support this work. The hope was that, in fact, by knowing these genetic mutations, we should be able to develop new kinds of therapy. The sad truth, the grim truth, is that, in fact, now, many years after this was initially accumulated, we're still wrestling with the consequences of these enormously complex data sets whose resolution we still don't really master. We don't really understand how they direct us in different kinds of therapy. And I say this with great sadness because it would be great if we understood the mutant alleles and how they created therapeutic sensitivities, how to treat such cancers. The fact is that advanced cancers now are not treated any more effectively than they were in general 20 or 30 years ago. In my own research lifetime, in 1979-80, my lab discovered the RAS oncogene present in human cancers. And now here we are 4 decades later, and this work has still not really accrued to the benefit of the cancer patient. So for one thing, to my opinion, sadly, I don't like to be a naysayer, we're a bit stuck in the mud right now in terms of being able to exploit these findings about the complex genetics of human cancer in terms of successfully implementing curative strategies. The second question was about single-cell RNA-seq, and single-cell RNA-seq is recently developed over the last 5 years and is a very powerful analytical tool in order to study the diversity, the heterogeneity of cells within a tumor. It's my opinion that the use of single-cell RNA-seq will begin to reveal in great detail how individual tumors actually develop. It may yield, although I'm not so sure about this, new ways of treating these tumors. But to the extent that we understand more and more about the pathogenesis, about the formation of human tumors, much of that understanding will come in the near future from doing single-cell RNA sequencing analyses, that is, understanding the transcriptomes of individual cancer cells that in aggregate create a macroscopic tumor. Does that help answer your question?
Thanks, Bob. I just want one more comment from you. So how do you see the single cell snapshots growing towards and contributing towards the study of temporal evolution of the tumors? So,
well, in principle, and it is now increasingly likely to be the case, one can use the single cell RNA sequencing studies to examine the precursor product relationships between different cell populations. How does one specific cell evolve or differentiate into another cell type? And so one can begin to plot out the developmental pathways of individual cells within a tumor, and on the basis of that, begin to understand the genealogical relationship between these individual cells and how they diversified and how one cell type evolved into the other. That obviously lies at the heart of understanding the complexity of the formation of a macroscopic tumor.
great, thanks, Bob.
My pleasure.
Okay, Dr. Faisal has his hands up. Dr. Faisal, you can go.
So thank you for excellent talk. I have a couple of questions. One of them is, you talked about epigenetic control of, epithelial to mesenchymal transition.
Yes.
So, I was wondering when does the tumor decide to go from epithelial to mesenchymal? A tumor is growing. What actually triggers this EMT? How does that happen? Number one. And the second is, when we're talking about tumor heterogeneity, are there cancer stem cells? Are there different populations of cancer stem cells as well? We know that a single tumor has multiple, genotypes within the same tumor, different mutations, Darwinian evolution as a result of that. But, are there only one type of cancer stem cells, or are there different populations?
So remind me what the first question is. I've already...
Sorry, the first question was what triggers the EMT?
Okay. And by the way, note that we're talking to one another and there's absolutely no delay in our conversation. Imagine here we are 7,000 or 8,000 miles apart from one another and we're talking as if we're sitting right next to one another. So that's quite miraculous to my mind. So the question is, what provokes the EMT? Cells do not spontaneously activate the EMT. They do so invariably in response to contextual signals they receive from other cells, in the case of carcinomas, from the reactive stromal cells, that is the cells like myofibroblasts and inflammatory cells in the stroma that release signals that induce the activation of a previously silent or latent EMT program in an epithelial cell or in a carcinoma cell. And this activation is, by the way, also reflective of what happens during wound healing when a normal epithelial tissue is wounded. The second question was, are there multiple stem cells within a tumor? I believe that epithelial cell populations naturally organize themselves into stem cells and non-stem cells. In other words, any population of epithelial cells, no matter how it's mutated or not mutated, naturally creates this hierarchical organization that's part of its behavioral repertoire. And therefore, I believe, I think it's likely to be the case, that as we go from one stage to the next stage of tumor progression, each of these populations of cells within a tumor at different stages of progression has its own subpopulation of stem cells. In an advanced tumor, which may have multiple subpopulations of cells coexisting within a tumor, I believe that each of those subpopulations of advanced tumor cells, once again, has its own flavor of cancer stem cell. So I believe that every population of stem cells, independent of their genetic aberrations, accumulates and encourages the formation of subpopulations of stem cells. I hope this helps to answer your question.
Okay, so, sorry, one last question is that, you know, the EMT cells that you showed were always present at the surface of the tumor when you showed the IHCs. So I
Yes.
was wondering whether they all develop at the surface or whether they migrate to the surface, once they become mesenchymal cells from epithelial.
Well, in principle, there's two alternative explanations. You've described the alternative one, which is to say that they originally started out with an EMT program, but at some distance from the stroma. However, one knows that by taking epithelial cells, both normal and neoplastic, and by applying to those cells the kinds of signals that are released by the stroma, including TGF-beta, notably, different kinds of non-canonical Wnt, IL-6, these signals coming from the stroma can induce a cell in a petri dish even to activate an EMT program. So indeed, it's really the actions of these heterotypic signals released by the reactive stroma and its associated cohorts of inflammatory cells that seem to activate the EMT program in previously epithelial carcinoma cells. So I think that's pretty well, that's on solid footing. In fact, I believe I showed you early on a cartoon of how a carcinoma cell can release interleukin-1 that impinges in turn on stromal cells, and the latter then reciprocate by releasing a whole series of EMT-inducing signals that ultimately elicit the activation of an EMT program in previously epithelial carcinoma cells.
The release of these EMT-inducing signals,
Yes.
does that depend on, size of the tumor, that, at particular size, it will only release the EMT-inducing factors from the stroma,
Well, in principle, it might be, but to my mind, it depends on the extent of tumor progression. As a tumor proceeds, the stromal cells become increasingly reactive. Fibroblasts become myofibroblasts. Macrophages and neutrophils are recruited into the tumor stroma. And so as the tumor stroma becomes more and more reactive, it becomes more and more capable of releasing signals that impinge on the carcinoma cells and evoke the activation of an EMT program in the latter. And by the way, all this recapitulates what the mechanisms that operate during normal epithelial wound healing. When epithelial wounding triggers a reversible EMT, where epithelial cells at the edge of wound sites turn on an EMT program, migrate to cover the wounded epithelial tissue site, and then after they've covered the wound site, will ultimately revert back to an epithelial state via an MET, via a mesenchymal epithelial transition, which therefore indicates the reversibility of the EMT program.
Thank you.
I have a question. Thank you, Professor
Madam.
Weinberg.
Can I ask a question?
Well, I think, do we have ladies first here, or how do we do this?
Yes,
Najma-
yes.
Absolutely.
Najma can ask.
Thank you, Professor Weinberg, for a very nice talk. So I had a question about on the basics that I know, that epithelial to mesenchymal transition takes place when the tumor tries to move from one place to another when it starts to metastasizing. But then when it colonizes to another place, and then it makes a tumor then and at that place. So what, and because in a 3D, a tumor, the cells which are residing in the inside of the tumor ball of cells, they are devoid of oxygen and other nutrients. So they may need oxygen and nutrients, for which they undergo increased angiogenesis, like the formation of blood vessels. So what triggers that angiogenesis?
Well, let me try to answer, Dr. Shaheen. First of all, something I didn't say is that carcinoma cells that have acquired an EMT, when they're in direct contact with the stroma, they can migrate into the middle of the tumor, away from the stroma, and may continue to express the EMT program, even if they're no longer in direct contact with the stroma. Secondly, when carcinoma cells leave a primary tumor and seed a distant tissue site, even though they may be more mesenchymal or quasi-mesenchymal at the moment of their arrival in that distant tissue site, eventually the bulk of those cells will undergo an MET, a mesenchymal epithelial transition, and thereby regenerate the hierarchy of stem cells and non-stem cells, of more mesenchymal and less mesenchymal cells in the distant tissue site. And in fact, if carcinoma cells that are fully mesenchymal or highly mesenchymal leave the primary tumor and seed a distant tissue, but they don't begin to generate more epithelial progeny via an MET, then those metastatic colonies will not grow very well. So the reversibility of this program is critical. None of this addresses your question about angiogenesis and how this intersects with the whole EMT, MET phenomenology. And one doesn't really know that much about it. It is the case that hypoxia can also activate the EMT-inducing transcription factor snail and thereby induce an EMT. But beyond that, I don't know how or whether the neoangiogenesis that occurs during the pathogenesis of an advanced tumor, how that occurs under the aegis of the EMT program. You may have noticed that among the long list of traits that I described that are acquired as a consequence of the EMT program, I did not list angiogenesis, simply because I just don't know the extent to which it's an important influence on the acquisition of blood vessels by incipient tumors.
Yeah, so one more question that I thought during the discussion, that as Dr. Faisal mentioned, that tumors have heterogeneous population of different types of cells. So I was assuming, or I was thinking, I was wondering how a single cell RNA sequencing, which is very popular these days, will help decipher the interaction among all these signaling. Because in a single cell, we will be having a single cell, and how would we be able to identify the exact signaling pathway that will lead to a 3D tumor?
Well, there's two ways to respond to your question. to the extent one is interested in the interactions between different cells coexisting within a given tumor, one needs to begin to describe what the individual cells look like, what they are phenotypically. And so the single cell RNA sequencing will allow us to begin to generate a catalog that enables us to identify the identities, the phenotypic identities, the differentiation identities of the individual cells within a given tumor. That on its own will not shed light on how these different potentially distinct cell types communicate with one another. That's a whole different challenge and is not resolved necessarily by single cell RNA-seq unless one learns from single cell RNA-seq whether those individual cells are releasing certain cytokines and growth factors and growth inhibitory factors. That may begin to shed light on the intercommunication between distinct cell types that co-reside within a given tumor. I fear I have to disappear in a short time, not from the surface of the earth, but from this Zoom conversation. I have a faculty obligation.
I have a question, Bob. I'm
Okay.
Tarek.
Yes.
So my question is regarding this epigenetic reprogramming
Yes.
in the stem cell. So I was wondering if the twist and snail led reprogramming of the cells, which involves this EMT, it
Yes.
follows the 5 mutations you talked about, like RAS, P53, etc., or this can happen in the absence of those genetic mutations.
Well, in fact, let's say we add one of these 5 mutations after the other, one, then a second one, third one, fourth one, fifth one, and one starts out in the beginning with a fully normal cell. Each of the cell populations that are created by the introduction of these oncogenes, for example, has the potential of activating its own EMT program, its own stem cell program. And so the EMT program operates in parallel to the activation of these different genetic states. Again, to repeat myself, both fully normal and highly malignant cells can activate their own or create their own subpopulations of stem cells. And so that kind of operates independently or orthogonally to the genetic alterations that these cells acquired during the course of multi-step tumorigenesis.
And just a quick question is about this twist and snail. You draw a wonderful parallel with the gastrulation movements in Drosophila, where tall signaling is involved early on, activating
Yes.
twist and snail. So do you have in this EMT, you think tall signaling will be involved there?
We've not seen much evidence of it. The fact is, at one point I mentioned in passing, there are multiple versions of the EMT program, and there are multiple distinct contextual signals that can activate the EMT program. And I don't know necessarily that toll signaling is one of these signals that can activate the EMT program in carcinoma cells. It may in other cell types, but not in carcinoma cells, to my knowledge.
Thank you.
Well, Mr. Jauhar, I can only say shukriya for the kind invitation. Thank you very much for having me. And inshallah, maybe one day I will come and be able to visit Lahore.
Inshallah.
Thank you so much for your
Yeah,
time. And once
Love to have you.
I think we can have a Western Punjab trip as well for you.
Oh, good. And maybe some good curries.
Yes, of course.
Bob, God bless you with a long and prosperous life, healthy life.
Oh, again, I say inshallah.
Inshallah. Thank
Thank
you
you.
very much. Thank you very much.
Thank you.
Bye-bye.
Bye.
Bye-bye.